All right, let's dive in. We're tackling nerve impulses today.
Ooh, exciting.
You know, I've been going through these notes you sent over from Dr. Rasha Eldeeb, and
Yeah.
wow, I mean, these tiny little electrical signals, right,
Yeah.
zipping around in our bodies, they're more complex than I ever imagined.
It's true. Once you start, like, really digging into how they travel, the speed, the different directions,
Yeah.
it really is mind-blowing.
Okay, so let's start simple. Unmyelinated nerve fibers, they're like, what? The back roads.
Yeah, I like
The
that.
back roads of our nervous system.
The scenic route.
Haha, exactly. Now, Dr. Eldeeb mentions local circular currents, which honestly, it sounds a little,
A little technical.
yeah, like
Yeah.
something straight out of a physics textbook. Can you help break it down?
Sure. Think of it like a chain reaction. You know,
Mhm.
so each section of the nerve fiber membrane, it gets activated.
Okay.
And that little activation, it triggers the next section, and then that one triggers the next, and so on and so on.
So it's like a domino effect.
Exactly. It's
Yeah.
self-propagating wave. And those local currents, they're what depolarize the next bit of the membrane, keeping that wave going.
So it's not a smooth flow.
Not really.
It's more like a cascade.
Yeah, a cascade of tiny little triggers, each one setting off the next.
That's actually a much cooler way to picture it. Now, here's where I got a little lost, I have to admit.
Okay.
Orthodromic and antidromic conduction. The impulse can travel in two directions.
Yeah, isn't that wild?
Why would a signal ever need to go backward? Like, what's the evolutionary advantage of that?
That's the million-dollar question.
Right.
So we know the orthodromic impulse, that's the one heading toward the axon terminal, right? That's the one that actually transmits the signal. But the antidromic impulse, the one going back towards the cell body,
Yeah.
we're still figuring that one out. There's some theories, of course. Some researchers think it might play a role in, kind of like fine-tuning.
Tuning, like,
Like regulating how excitable the neuron is. Or maybe it's a feedback mechanism, giving the neuron information about the strength of the signal it just sent out.
so a little quality control check.
Possibly. It's still a big mystery, but it shows you just how intricate neural communication really is.
It really does. Okay, let's switch lanes now. Let's talk about those soupy highways, the myelinated nerve fibers.
Ah, yes, those are the fast ones.
They've got that myelin sheath, which basically acts like insulation, right?
Exactly. Think of electrical wires in your house.
Okay, yeah.
You don't want to just bare, right?
Yeah.
The insulation prevents shorts and makes sure the electricity flows where it needs to. Myelin does the same thing for our nerve impulses. Keeps things running smoothly and super fast.
Never thought about it like that. So Dr. Eldeeb uses the term salutatory conduction. Is the signal actually like jumping?
In a way, yes. Instead of traveling along the whole axon, inch by inch, the action potential, it leaps between those gaps in the myelin.
Gaps.
Yeah, they're called the nodes of Ranvier.
Nodes of Ranvier. Got it.
And this jumping, it's way more efficient.
Yeah.
Faster and less energy wasted.
So instead of walking across a room, it's more like hopping across it.
Uh-huh. That's a great way to put it.
You get there faster and you don't get as tired. And because the signal only has to regenerate at the nodes,
Right.
that saves a ton of energy.
Exactly. Brilliant design, really.
Our bodies are so smart. All right, let's shift gears a bit. How do scientists actually record these signals, these action potentials?
Ah, yes, getting into the nitty-gritty.
Dr. Eldeeb's notes mention biphasic, monophasic, and compound action potentials. Seems like a lot of ways to measure the same thing.
Well, each method gives you a different perspective. It's like looking at a sculpture from different angles. With biphasic recording, you've got two electrodes. They're both on the outside of the nerve fiber.
Okay.
And as the impulse passes, you see, like, an upward deflection, then a downward one. It's like capturing the peak and trough of a wave.
Okay, I can picture that. What about monophasic recording? How's that different?
Monophasic, you're essentially looking at the signal from just one side. So one electrode is inside the fiber, one's outside, or maybe one's over a damaged part of the membrane.
So you're getting, what, a closer view?
Yeah, like a close-up of one side of that wave, not the whole shape.
Oh, I see. And then there's compound action potentials.
Right, those are the complex ones. Imagine you're listening to a whole choir, not just one voice. Each singer has a slightly different pitch and volume.
Okay, yeah.
That's what a compound action potential looks like. You're picking up on all the different fiber types within a nerve, each with its own speed, and it all blends together into this complex waveform.
That makes sense. Now, speaking of different fiber types, Dr. Eldeeb mentions a classification system, group A, B, and C. Scientists really love their categories, don't they?
Organization is key, especially when you're dealing with something as intricate as the nervous system. This system, It's all about fiber diameter and how fast the signals travel. Group A, those are the heavy hitters, the largest, the fastest. We're talking speeds up to 120 meters per second.
Wow, that's like race car speeds.
Exactly. They're the ones responsible for those lightning-fast reflexes, precise muscle control, all that good stuff.
Impressive. What about groups B and C? Are they just slower versions of group A?
Not quite. Each group, it has its own specialty. Group B fibers, they're the middle ground. They're important for the autonomic nervous system. So think heart rate, digestion, things like that.
Okay, so more like the steady, reliable workers.
Exactly. And then you've got Group C, the smallest and the slowest. They handle things like pain signals, temperature, and regulating blood flow to the skin.
So it's not just about speed, it's about specialization. Each fiber type has its own specific job to do.
You got it. And even their vulnerabilities are different. Group A fibers, they're more susceptible to pressure. That's why you might get that numb or tingly feeling if you sit funny for too long.
Oh, that makes sense. So it's like you're literally putting the squeeze on those fibers.
Uh-huh. Yeah, kind of. Group B fibers, they're more sensitive to oxygen deprivation. Makes sense, right? Heart rate, digestion, those need a constant supply of oxygen to work.
Absolutely. And group C.
They're the ones that are easily blocked by local anesthetics.
So that's how they numb you up for a procedure.
Precisely. They target those C fibers so you don't feel the pain.
Wow, it's amazing how each fiber type is so perfectly adapted. It's like a whole team of specialists working together. Now, before we move on, there's one more thing from Dr. Eldeeb's notes that I want to touch on. Electrotonic potentials. What are those exactly?
Yes, those are the subtle ones. It's like a whisper of activity, even when a signal isn't strong enough to trigger a full-blown action potential.
So even a weak signal can have an impact.
Exactly. It's like the neuron is always listening, even to those quiet whispers. And we have two main types, anelectrotonic and catelectrotonic.
Okay, and what's the difference?
Well, anelectrotonic potentials, they actually make the neuron less likely to fire.
Really? So even though they're not triggering a signal themselves, they can actually stop one from happening.
Exactly. It's a way of dampening things down, keeping the neuron from getting too excited.
And catelectrotonic potentials, those are the opposite.
Right. They nudge the neuron closer to that firing threshold, making it more sensitive.
So it's like a constant balancing act between these two forces.
Exactly. A constant push and pull, shaping the flow of information through the nervous system.
This is all so incredible, and we've only just scratched the surface. I can't wait to dive even deeper into the world of nerve impulses in the next part of our deep dive.
Me too. There's so much more to explore. So those electrotonic potentials, the ones that don't quite make it to a full action potential, they're not just, background noise.
That's what I was thinking. They seem pretty subtle. Do they really make a difference?
Oh, they can. Take anelectrotonic potentials, for instance.
Okay.
They can actually cause something called anedyl block. Essentially, it's like putting the brakes on nerve impulse transmission.
Wait, really? So even though they're not triggering a full signal,
Right.
they can actually stop one from happening.
Exactly. It's all about the membrane potential. Anodal currents, they increase that potential, making it harder for the neuron to reach that threshold to fire. Kind of like dampening its excitability.
Interesting. So on the other side of the coin, you have the catelectrotonic potentials, right?
Right.
And those, they bring the neuron closer to firing.
Precisely. It's like giving it a little boost, making it more sensitive to any incoming signals.
So it's this constant tug of war between these two forces,
Exactly.
shaping how information flows through the whole system.
Exactly. It's amazing, isn't it?
It really is. But how does all of this tie back to those different nerve fiber types we talked about? Dr. Eldeeb mentioned that some are more vulnerable to pressure or oxygen deprivation or even anesthetics.
That's a really important point. It's not just about how fast a signal can travel. It's about how tough each fiber is, how resilient they are to different kinds of stress.
Okay.
Take those group A fibers, the super speedy ones responsible for all those quick reflexes. They're actually pretty sensitive to pressure.
Which seems kind of counterintuitive, right? You'd think something that needs to react so quickly would be more, robust.
You'd think so, wouldn't you? But their larger diameter, it might actually make them more susceptible to that anodal block. So even a little bit of pressure can mess with their ability to transmit signals.
So maybe that's why your leg goes numb if you sit on it funny for too long.
Could be. You're basically squeezing those group A fibers, making it harder for them to fire properly.
Wow, that makes a lot of sense. Okay, what about group B fibers?
Right. Those are the ones involved in the autonomic nervous system.
Right. Heart rate, digestion, all that.
Exactly. And they're particularly vulnerable to oxygen deprivation.
Which again makes sense, right? Those functions need a constant supply of oxygen to keep going.
Absolutely. And their sensitivity to oxygen might be related to their reliance on those catelectrotonic potentials.
Yeah.
Remember, to maintain that balance, to be able to boost their excitability when needed, it takes a lot of energy. And that energy comes from oxygen.
So if they don't get enough oxygen, they can't keep up that delicate balancing act.
Exactly. It's like a car running out of gas. It just can't function properly without that fuel.
That's a great analogy. Okay, last but not least, group C fibers. The slow and steady ones involved in pain and temperature. Dr. Eldeeb mentioned that they're easily blocked by local anesthetics.
That's right. And that actually makes sense from a clinical perspective. You want to be able to block those pain signals without shutting down everything else, right?
Right, of course. But why are they so sensitive to anesthetics? Is it something about their structure?
It could be related to the types of ion channels they have or the properties of their myelin sheaths. There's still a lot we don't fully understand, but it's an area of active research.
Fascinating. It's just amazing to me how much variation there is, even within something as seemingly simple as a nerve fiber. Each type has its own personality, you could say, its own strengths and weaknesses.
Absolutely. And understanding those nuances, that's key, not only for appreciating just how complex the nervous system is, but also for figuring out how to treat neurological disorders more effectively.
Because if you know how a particular type of fiber is likely to malfunction,
Right.
you can tailor the treatment to address that specific vulnerability.
Exactly. For instance, in multiple sclerosis, the immune system attacks the myelin sheath, and that disrupts that speedy salutatory conduction we talked about earlier.
Oh, right, right. So it's like stripping the insulation off those electrical wires.
Exactly. And the symptoms, they can vary so much depending on which nerve fibers are affected. If it's mainly motor fibers, you might see problems with movement, coordination. If it's sensory fibers, you might get numbness, tingling, even pain.
So it's not a one-size-fits-all kind of disease.
Nope. The specific symptoms really depend on which type of fiber is being targeted.
Wow. So understanding these different fiber types, their vulnerabilities, it's all connected to the bigger picture of how the whole nervous system functions.
Exactly. It's all interconnected.
This is all so fascinating, but I have to admit, it's a lot to take in. There's just so much going on, so many intricate little mechanisms working together.
It's true. The nervous system is incredibly complex, but that's also what makes it so exciting to study. There's always something new to discover, some new puzzle to solve.
And each new discovery brings us closer to understanding how our brains work, how our bodies work, and ultimately, how to help people who are struggling with neurological disorders.
Absolutely. So as we move into the final part of our deep dive, let's shift our focus to the synapse, that incredible meeting point between neurons,
Okay.
where those electrical signals we've been following, they get transformed to chemical messages.
Because it's not just about the signal traveling down the fiber, right? It's about how that signal gets passed on to the next neuron in line.
Exactly. And that's where things get even more fascinating. We'll be diving into the world of neurotransmitters, receptors, all those intricate mechanisms that control how neurons talk to each other.
I can't wait. It sounds like we're about to uncover a whole new level of complexity in the world of nerve impulses. Okay, so we've traveled the length of the nerve fiber, we've seen all the ins and outs, and now we've arrived at the synapse. It's like the grand finale of nerve impulse transmission,
It
right?
really is. It's where everything we've been talking about, all those electrical signals, they get translated into this whole other language, the language of chemical messages.
So it's how neurons actually communicate with each other.
Exactly.
I mean, it's like they need a translator, right, to understand each other.
That's a great way to put it. And those translators, they're called neurotransmitters.
Okay.
These tiny little molecules, they carry the signal across that gap between neurons.
The gap, that's the...
The synaptic cleft.
Right, the synaptic cleft. Dr. Eldeeb, she describes this whole process like a carefully choreographed dance.
It is. There are so many moving parts.
So walk me through it. What actually happens when that electrical signal, that action potential, reaches the end of the line?
Okay, so picture this. The action potential arrives at the axon terminal.
Okay.
That's the end of the presynaptic neuron, the one sending the signal. And when it gets there, it triggers these voltage-gated calcium channels to open.
Hold on, voltage-gated, like the ones we talked about with action potential.
The very same. They're like tiny little gates in the membrane, and they only open when there's a change in the electrical charge.
Oh, got it.
So these calcium ions, they flood into the neuron, and that influx, that's the signal, It tells these little packages called synaptic vesicles to fuse with the membrane.
Synaptic vesicles, those are the ones that hold the neurotransmitter.
Exactly. They're like tiny little bubbles filled with those chemical messengers. And when they fuse with the membrane, it's like whoosh, all the neurotransmitters spill out into the synaptic cleft.
It's like a burst of confetti.
I love that.
Okay, so now we've got all these neurotransmitters floating around in the synapse. What happens next?
Well, on the other side of the cleft, the postsynaptic neuron, the one receiving the signal, it's covered in these special proteins called receptors.
Oh, okay.
And each receptor, it's designed to bind to a specific neurotransmitter, like a lock and key.
So each neurotransmitter has its own designated docking station.
Exactly. And when they bind, that triggers a change in the postsynaptic neuron.
It's not just a passive thing. It actually causes something to happen.
Right. The binding can either depolarize the postsynaptic neuron, which brings it closer to firing its own action potential,
Okay.
or it can hyperpolarize it, making it less likely to fire.
So even at the synapse, we've got that same interplay of excitation and inhibition.
Exactly. It's all about balance. And the type of response, it all depends on which neurotransmitter and which receptor are involved. Some neurotransmitters, they're mainly excitatory, like glutamate. Others are mostly inhibitory, like GABA.
So the same signal can have different effects depending on where it ends up.
Precisely. The context matters. And to make things even more interesting, we also have neuromodulators.
Neuromodulators.
Yeah, these are molecules that can, like, fine-tune the whole process.
But they're like what, the control knobs on a soundboard? They can adjust the volume of the signal?
That's a great analogy. They can make a synapse more or less sensitive to a particular neurotransmitter, kind of like adjusting the volume, exactly.
It's like a whole symphony going on. All these signals, modulators, receptors working together.
It really is. And just like an orchestra, things can go wrong if the balance is off. Too much excitation, you might get seizures. Too much inhibition, you might get lethargy, even a coma.
So finding that sweet spot, that balance between excitation and inhibition,
That's the key.
that's what keeps everything running smoothly.
Exactly. And it's a dynamic balance. It's constantly being adjusted based on all sorts of things.
Yeah.
Genetics, development, experience, environment. It's incredibly complex.
This whole journey has been incredible. I mean, we started with these tiny little electrical signals, and now we've uncovered this whole world of chemical messengers and this incredible dance of regulation at the synapse.
It's mind-blowing, isn't it? All of this is happening inside us right now. It's what allows us to think, to feel, to do everything we do.
It's a reminder that there's so much more to us than meets the eye, and we're still just beginning to understand it all.
Exactly. What we've talked about today, it's just a glimpse into this vast, ever-evolving field of neuroscience.
But it's a crucial one, right? I mean, this knowledge, it's the foundation for understanding how our brains work and what goes wrong in all these neurological disorders.
Absolutely. The more we learn about nerve impulse transmission, the better equipped we are to treat conditions like Alzheimer's, Parkinson's, multiple sclerosis. The list goes on and on.
It's like we're unlocking the secrets of the brain, one synapse at a time.
I love that.
Well, I think that brings us to the end of our deep dive. But this is definitely a topic that I'll be thinking about for a long time. It's incredible to think that something as seemingly simple as an electrical impulse can give rise to such complexity and beauty.
It really is. It's a testament to the power of the human brain. And as we continue to explore its mysteries, I'm sure we'll uncover even more wonders.
Well, to our listener, thank you so much for joining us on this incredible journey into the world of nerve impulses. We hope you've enjoyed it as much as we have. Until next time, keep those synapses firing.